Click for IB Biology. Here we're looking at Endosymbiont Theory. There's a blank PDF of this presentation down in the description, and there you'll also find a link to 10 questions with answers that you can use to test your knowledge and understanding of endosymbiont theory. This theory was published in 1967 by the American scientist Lynn Margulis. The basis of this hypothesis is that when we look at complex eukaryotic cells, we see within them structures like mitochondria or chloroplasts. The nucleus would be included in this discussion. And Margulis' hypothesis was that these complex eukaryotic cells owe their origins, which is about 2 billion years ago. The origin of these complex cells came about as different prokaryotic cells combined their cellular processes into a single functioning unit. This is the nature of the symbiosis, where we have a cooperative strategy for improving their efficiency, certainly within an evolutionary concept. And Margulis argued that mitochondria and chloroplasts were two particular examples, and indeed the nucleus are examples of this kind of symbiosis, cooperation between different types of prokaryotic cell to a higher level of functionality and a higher competitive edge in the natural selection process. Her evidence came from things like comparisons of ribosome structures, DNA structures, reproductive strategies, biochemical pathways, and modes of protein synthesis. Let's take a look at one example to see if we can appreciate the symbiosis, but also the endosymbiotic part. Now, students will find this explanation much more accessible once they have studied respiration, and so I advise that you return to this endosymbiont video at that time. But let's consider in the 2 billion years ago that there was a cell, we'll call this cell A, and its function, its ability was to take 6 carbon sugars. So C6 refers to sugar. Perhaps students know C6H12O6, the classic glucose molecule, and they're able to take that in from the environment. And they have enzyme structures within their cell cytoplasm to break down this 6 carbon sugar. Imagine 6 carbons in a row, and they can break it apart, and in doing so they release energy enough to synthesize a small amount of ATP. The end of the breaking apart is to leave C3 compounds. So if we take C6 and we have an enzyme which snaps this molecule in half, we would end up with 2 C3 molecules and a little bit of ATP. So that sounds great. The organism will regard the C3 sugar as being waste, and so will excrete it into the environment. In the same environment, there's another cell, let's call this one B, and it takes C3 sugars into its cytoplasm, uses an enzyme system to gradually break those down to single carbon structures, carbon dioxide, and of course it will then excrete that as a toxic waste. But in this process, which is enhanced by consuming oxygen, it makes a great deal of ATP. So Margulis' argument was that these two cells, A and B, decided to combine their chemistry. And so within a single cellular structure here, shown here, this is the symbiotic cell, a combination of two. The large cell takes in C6, much like we have here, and breaks it down to C3. Now, instead of excreting this, it passes it directly to the second cell, which then is able to break down the C3 to single carbons and with the excretion of the carbon dioxide. Of course, to do this efficiently, it requires oxygen. So the symbiotic relationship is that the B component obtains C3 in large quantities and easily as they are provided directly. The first component, the A component, receives the additional ATP, a share of that ATP, which is retained within the cell and used by this single cellular structure. So the symbiosis is that we arrive with a more efficient way of breaking down the sugar in which the two original cells benefit equally, and the single cell formed is much more efficient. Remember, this is called Endosymbiont Theory, and the endo part refers to the Margulis hypothesized that, let's retain my AB feature, cell A engulfed cell B to create this single structured, let's call this cell C, this one here, like this. And so then the B cell unit now becomes in here like so. But remember, it would have the two membranes around it, the one from cell A, but also its own. This can be thought of as a mitochondria. And the evidence shows that the mitochondria and chloroplasts have their own DNA, which is very prokaryote-like. Their protein synthesis mechanisms are very prokaryote-like. Their ribosomes look like prokaryotes, very different to the ribosomes and other structures which are found in the cytoplasms of the host cell. So this is called endosymbiont theory. The same argument is used to explain things like the nucleus and also chloroplasts. So you might want to see if you could look up and find descriptions of their evolution. Endosymbiont theory, Lynn Margulis, the combining of prokaryotic cells about 2 billion years ago to create the complexity of eukaryotic cells, a major step forward in the evolution of living things.